And did you receive the data pack my colleague sent through to you?
Yes, I have it in front of me, so I made a print of it. Yeah.
Thank you. So today we'll be discussing your opinions on the phase 3 data for filgotinib and upadacitinib presented at EULAR 2019, the safety of JAK inhibitors in immunology, your opinions on the development of otilimab in RA, and then perceptions of current phase 2 candidates and their mechanisms of action. So jumping in with filgotinib, there's data from the phase 3 FINCH 1 and FINCH 3 studies in slides 2 and 3 of the data pack. These data were first announced in press releases in March of this year and data presented in late breakers at EULAR 2019. So if we look at FINCH 1 to start with on slide 2 of the data pack, what is your opinion on the efficacy of filgotinib in this patient population?
So I think overall that the phase 2 data were published in ARD a couple of years ago, and I think that the phase 3 trial data, I saw them also at EULAR, looks promising. You have, for example, the FINCH 1 study. You have some higher response rates with a higher dose compared to 100 milligram. And also higher with 200 milligrams compared to the active comparator, adalimumab. So, and whether you have the same concern with filgotinib as you have with tofacitinib and baricitinib regarding the dose and cardiovascular events, or thrombotic cardiovascular events, I don't know, but I just noticed that the higher dose seems to be at least numerically superior to the 100 milligram dose, at least in FINCH 1, and I think it's more or less the same in FINCH 2. Sure.
And do you have any thoughts on the active comparator comparison here, so versus adalimumab?
Yeah, so that's of course always interesting that when you have a superiority compared to the active comparator, and here also the number of patients in adalimumab arm was pretty high. And it was also superior to placebo. But again, the ACR 70 and 50 responses, for example, are very similar with the 100 milligram filgotinib dose and the 40 milligram adalimumab dose.
Right, okay. So if we move on to the FINCH 3 data now on the next slide of the data pack, what's your opinion of filgotinib's efficacy in the methotrexate naive patient population?
Yeah, so it's, that you have, like with the filgotinib 100 milligram dose, that you have, ACR 20, 50, and 70, like 80, 58, and 40. That's good. That is in combination with methotrexate. If you look at the monotherapy, so then, numerically, again, filgotinib 200 milligram in combination with methotrexate is slightly superior to filgotinib monotherapy.
Right. And do you have any comments on the comparison to placebo for the monotherapy, looking maybe specifically at the ACR 20 not reaching statistical significance versus methotrexate?
Yeah, so that's of course a bit surprising, because you always think that the IL-6 inhibitors and the JAK inhibitors, they are usually as monotherapy superior to methotrexate. So that's a bit surprising in this particular study, and I don't know exactly why there is no difference between the JAK1 inhibitor and methotrexate.
Right. Okay.
So so there is, as you say, for ACR 20, yes, but for ACR 50 and 70, you have superiority.
Sure. So does that maybe impact your opinions on the efficacy of the monotherapy, or do you still think that's maybe the potential for an effective dose?
Pardon? Once more.
Does the ACR 20 not reaching superiority, but then the ACR 50 and 70 showing superiority, does that impact your view of the monotherapy efficacy of filgotinib?
No, actually not.
Okay.
So because ACR 20, that's not a clinically important endpoint. It's more like a regulatory endpoint, whereas for clinical use, you're more interested in aiming for ACR 50 or ACR 70.
Great, thank you.
And also DAS28 remission.
Sure.
Yeah, and you also see that there is a numeric, these are methotrexate naive patients, so that means that they probably have a rather short disease duration, and that means that you have very little radiographic damage, and you have just a numeric difference for radiographic non-progression.
Right, sure. So, in your opinion, from the studies you've seen here, what is what is the optimum dose for filgotinib and RA?
That is a very important question, and I'm not sure. So, from an efficacy point of view, it seems to be 200 milligram for safety point of view. You haven't so much safety data here, but
So that's later in the day.
I think I remember. What? You're coming back to that
safety data, which we'll get onto in a couple of questions.
Yeah. So it's. I'm not sure, actually. Gilead and Galapagos, which dose they go for when they have their regulatory application, but so I don't remember if they really go for 100 or 200 milligram.
Right. Would you like to see both doses available in the clinic?
I think so, especially if they have flat pricing.
Right.
That's always a problem when. If a company come out with a lower dose, like a 100 milligram, and then they say that, okay, you use 200 milligram, then you need 2 tablets of the 100 milligram, so you should have tablets both with both doses and preferably with flat pricing.
Okay, thank you.
Because otherwise, the 200 milligram dose will be extremely expensive.
Right, yeah, that makes a lot of sense. So I'm going to ask about some indirect comparisons for filgotinib versus the other JAK inhibitors. Hopefully this isn't too repetitive, but if it is, feel free to just say so. But if we can first look at the FINCH 1 study, do you have any thoughts on how the efficacy for filgotinib from this study compares to that seen with upadacitinib, baricitinib, and tofacitinib?
So I think my overall impression when I looked at the 4 JAK inhibitor phase 3 data is that there is no major difference. In the BEAM study with baricitinib, you have superiority for adalimumab. Here in FINCH 1, you have also some numeric superiority, and at least for the 200 milligram dose, as we talked about. Mm-hmm. And I know that for UPA, okay.
Yeah, that's fine.
You also have some superiority, but for tofacitinib, you don't have superiority, like in the STATUS trial. So I think my impression overall is that if there is a difference, it's always difficult to compare different studies with each other because some of the baseline characteristics and that will be slightly different. But my impression is that overall there is no clear difference between the different JAK inhibitors.
Okay. And would that also carry through for an indirect comparison of the FINCH 3? So looking at in early RA or with the monotherapy?
Yeah, I think so. When you have, it's pretty good to have, like with the 100 milligram dose, that you have 80, 58, and 40% ACR 20, 50, and 70, which is that's pretty good.
Right.
Yeah.
Sure. And then sort of a hypothetical question, just to round off this, is if filgotinib were to be approved for the treatment of RA in Europe, where do you think that would fit into the treatment algorithm?
That depends very much on the price.
Right. Okay.
In different markets. I'm coming from Norway, so we have we have extremely good access to both targeted synthetic DMARDs and biologic DMARDs, but also the health authorities are very much focused on the cost. So every year we have a national tender, and then now, are you aware of the price of Humira in Norway now?
Not off the top of my head.
No, it's €2,000 per year.
Oh, right. Okay. So,
So then you understand. Yeah. And we have had biosimilar infliximab on the market since 2014, starting out with 39% lower price than Remicade, and next year 69% lower price. So the budget for all these agents are in the healthcare system. So for me as head of the department, I have the budget for these drugs for the population served by my hospital. So which means that we have a very strong focus on cost issues. So it depends very much. The positioning of filgotinib will very much, at least in this paradoxically, in the richest countries, it will very much depend on the price.
Right. And if pricing was in line with other JAK inhibitors, do you think it would just be another JAK inhibitor, or is there any difference in maybe targeted positioning? Or no comment, that's fine as well.
So compared to the other JAK inhibitors?
Yeah.
You said?
Yeah, I mean, do you think there's a particular patient population where this might be more suited for uptake?
More treated, you said?
I was asking if there's a specific patient population where you think that filgotinib could see more rapid uptake if it were approved. Not compared to other JAK inhibitors, but the patient population where the JAK inhibitors, in my opinion, and I think that's also in line with the EULAR recommendations for using synthetic and biologic DMARDs, that is that when patients are intolerant to methotrexate, both IL-6 inhibition and JAK inhibition seems to have some superiority compared to the TNF inhibitors, for example. Okay, thank you. Very helpful. So now moving on to some safety for JAK inhibitors in immunology. I guess starting off with just a general question, what are your main safety concerns when using JAK inhibitors in your clinical practice, or that you've identified from clinical trial experience?
So I think I need to focus on clinical trial data, because there are not so much. We have some European collaboration on JAK inhibitors now with registries, but we don't have so much data yet. Something submitted to ACR from this collaboration, but so I think that we need to see more real-life safety data before we really know more about the clinical importance or relevance of the thrombotic cardiovascular events. We know a lot about, infections and herpes zoster, and that differs also in different parts of the world. It's more in Asia than in Europe, as far as I know. So I think it's infections and the cardiovascular issues, and maybe also lipid levels. We need more safety data from the real world, and we need to learn more about how we appropriately should monitor these patients with regard to lipids and the neutropenia and liver enzymes and all these things.
Very helpful, thank you.
But overall, I don't think JAK inhibitors raise any more concerns than biologic DMARDs, but there are some different profiles regarding safety issues. It seems to be so.
Good. Okay, thank you. So now if we look on to slide 4 of the data pack, this is some integrated safety analysis from the SELECT program for upadacitinib through to about 1 year of exposure. So what are your thoughts on the safety profile of upadacitinib as observed in this integrated analysis?
Yeah, so if you look at these, data, for example, that you have in the table here, there is no major difference between adalimumab and UPA. And for example, for so VTE, that is the venous thrombotic event. Is that correct?
Yes. Yeah.
So and overall MACE, there is no real difference. But again, here also for 30 milligram with UPA, you have some more adverse events than with the 15 milligram, and I know that for the efficacy data, 30 milligram is slightly superior to 15 milligram, and I think that AbbVie, they focus on 50 milligram as a primary dose in rheumatoid arthritis, as far as I know.
Mm-hmm.
Because you see, for example, herpes zoster, you have twice as many events with herpes zoster with the 30 milligram compared to the 15 milligram, and it's higher than for placebo and for compared to methotrexate and also adalimumab, but for the 15 milligram, there is no difference.
Right. Okay.
No, so that was that was serious infection. But for overall for herpes zoster, you have more with UPA 15 milligram, numerically more than for the other comparators, and 30 milligram more than for 15 milligram.
Okay. And how do you think the safety data compares to that available for baricitinib and tofacitinib? Is there anything that sort of stands out as different here?
Oh, I don't think so, and we need to see more real-life data and more pharmacovigilance data to really know whether there are any major difference. And again, it's difficult to compare phase 3 trials because of some differences in the patient populations. Okay.
Thank you for that. Earlier this year, both the EMA and FDA highlighted an increased rate of pulmonary embolism and death from a phase 4 safety study of tofacitinib in the 10 milligram twice daily arm. There's some background about this on slide 5 of the data pack. So how does this observation impact your perception of tofacitinib for the treatment of RA, if at all?
So can you say, what was the endpoint? You said that 10 milligram was more frequent than 5 milligram. Well, it was
pulmonary embolism and death.
Yeah, exactly. So it's the same as for baricitinib, because I think that the first concern was raised with baricitinib 4 milligram when FDA evaluated their submission package, and then later they also reviewed the tofacitinib 10 versus 5 milligram. Mm-hmm. So again, I think that also the regulatory agencies, they have raised concerns about the higher dose of 10 with the 10 milligram tofacitinib. So I think that the impact is that it will be very hard to use the high dose of tofacitinib in particular.
Right. And are there any patient groups in RA that the 10 milligram twice daily dose is currently useful to your knowledge?
No, I think my understanding is that the 10 milligram dose is more relevant for seronegative conditions like maybe spondyloarthritis in the future and psoriatic arthritis also currently. Okay.
And has this information altered your prescribing behavior for tofacitinib?
No, it has not, because I think that for 5 milligram twice daily, there is no major concern. And again, in Norway, last year, tofacitinib was less expensive than baricitinib. Of course, the patients then prescribed tofacitinib, they will continue with tofacitinib. But this year, baricitinib has a price which is less than tofacitinib. So baricitinib is the preferred JAK inhibitors currently in Norway based on this, because we consider them similar regarding efficacy and safety.
Okay, very helpful, thank you. And then more broadly now, has this safety announcement impacted your view of the safety of the JAK inhibitor class as a whole?
No, except as we discussed also for the JAK1 inhibitors, that there are some concerns with the dose-related occurrence of certain adverse events, which may be both infections and thrombotic events. So I think that there are some caution raised with a higher dose, and in particular in patients who might have some predisposition for thrombotic events based on their disease history or concomitant medications or whatever.
Okay. So now if we move on to the next slide, we're looking now onto some safety data for filgotinib. So starting off with pooled week 24 data from the phase 3 trials. So what is your opinion of the week?
Just a second. So the slide now, that is, what is the Is that the Gilead and
Gilead
Galapagos
and Galapagos report, updated safety
Yeah.
information, and it's the week 24 data.
Yeah.
Okay, so what is your opinion of the week 24 safety data from the phase 3 FINCH program?
Yeah, so here they pooled the 3 FINCH studies.
Yeah.
So, So again, if you look at, the adverse events of particular interest, if you look at herpes zoster, so, the percentage is not overall different from adalimumab. If you look at pulmonary and venous thrombotic events, also the same, no difference. That's also, the numbers are so small.
Mm-hmm.
So, but I don't think, these pool data are actually very promising for filgotinib. But again, I would have liked to see a comparison. Let me see if that is on the next one. No. So I would have liked to see a comparison of the 200 and 100 milligram dose.
I mean, so there are the 2 doses there for the 100, but obviously, the 100 and 200 milligram, it's just laid out slightly differently to the other tables.
Yeah.
Right.
Sorry. I looked at the total at the bottom.
Oh, right.
Okay. But here, for you are right, that 100 and 200 milligram does not seem to be different from each other, which is, of course, very positive since we also looked at the 200 milligram dose was numerically slightly superior regarding efficacy. So based on these data, there doesn't seem to be any major dose-dependent relationship to adverse events with filgotinib. At least that's my immediate interpretation from this data.
Okay. And I mean, maybe this is a bit speculative again, but if you were to start a patient on filgotinib, would you start, based on these data, with the 100 or the 200 milligram dose?
I'm not sure, but
okay.
It seems that if you look, on the balance, if you really go for efficacy, and that's why you start with these drugs, and when you looked at the 200 versus 100 milligram dose in the first slides, and look at the safety information here, it seems that 200 milligram would probably be the preferred dose.
Okay, thank you. And then if we move on to the next slide, this is data from week 156 from a phase 2b long-term extension. What are your thoughts on this data?
Again, that is the phase 2 data trials and Darwin studies that were published in ARD. And again, here you see that the percentage of patients with thrombotic events and the patients with cardiovascular events is pretty low. And serious infections also pretty low. Herpes zoster 1.5%, which is not so bad.
Okay. And does the safety data we've seen for filgotinib impact your view of the safety of the JAK inhibitor class as a whole, or not?
No, I don't. It seems immediately when you look at this data that filgotinib may have a slightly better safety profile than the others, but again, as I said a few times, that it is very difficult to compare studies, and especially to compare phase 3 data with phase 2 data. That's because the patient populations are usually very different.
Sure. And do you think there is any difference in the safety profile of JAK1 specific inhibitors versus broader targeting JAK inhibitors?
No, I don't. I think overall it seems to be more or less the same, because you have also an elevation of herpes zoster here, more or less the same as you had with tofacitinib and baricitinib. But again, it depends very much on which countries recruited patients to different studies and patient populations overall and trial designs.
Okay, and then if we look onto the next slide, there is some updated safety information from 7 years of treatment with baricitinib. Do you have any thoughts on this data? Or,
yeah. So here you see this if you look at the thrombotic events, that if you compare, since the dose, of 2 versus 4 milligram is, of particular interest now because of the FDA opinion. And then you see here that you have a higher 1.3% versus 0% with the 4 milligram versus 2 milligram dose for deep vein thrombosis and pulmonary embolism. So when you say the 2 milligram to 4 milligram extended, that is the patients who were originally on 2 milligram and then went on with 4 milligram, I assume?
No, so it's patients either continuing on 2 milligram or 4 milligram through the rest of the study, open label.
Okay, yeah.
But so I think that's all. I think the new data is in the right-hand most column, but that's just pooled data for baricitinib, I believe. Maybe.
Yeah. Okay, so I'm not sure, but I think that it's in some way it supports that at least numerically the numbers are small, but it supports that there is a dose effect for deep vein thrombosis and pulmonary embolism for 4 milligram versus 2 milligram. So that is consistent with the view of ACR, whereas EMA didn't pick that up and baricitinib was approved in Europe. Mm-hmm. But again, I think that in clinical practice, you should apply some caution, especially in patients at risk of pulmonary embolism, and maybe then consider 2 milligram instead of 4 milligram. But the efficacy data is much more convincing on 4 milligram than on 2 milligram.
Right, sure. Okay, so now we're going to move on to looking at some data for upadacitinib from week 48 or week 60, which was presented at EULAR. So there's data from all 5 of the studies which was used for their global filing. So these are on slides 9 through 13. But I'm just going to go through them sort of as they are in the data pack. If you have any thoughts on those, it would be very appreciated as we go through them. So if we could start off with Select Early, which is on slide 9 of the data pack.
Yeah.
Do you have any thoughts on this data?
So it's also again, UPA 15. So just a second. So that was this was the patients. Yeah, exactly. So these patients, they were methotrexate naive, I think.
Yes.
Yeah. So they were methotrexate naive and were randomized to methotrexate versus 50 milligram versus 30 milligram monotherapy with UPA. Mm-hmm. And here you see that, if you look at ACR 70, for example, the response rate is nearly twice as high with upadacitinib compared to methotrexate, which is good. So again, supporting what I mentioned before, that especially important population for using JAK inhibitors is the population who cannot use methotrexate as co-medication.
Right, sure. Okay, and if we can move on to the next slide, this is data from Select Compare, including the comparison, obviously, to adalimumab. Do you have any comments on this data?
So here you have, with 15 milligram in combination with methotrexate, you have 36% ACR 70, you have DAS 28 remission 38%, and the active comparator arm with adalimumab, it's 23 and 28. So of course this is good and it's statistically significant. So these data I think were first presented at ACR last year. I think so.
So this is new data now at a later time point, so week 48, whereas we'd only seen, I think, previously week 24 maybe data. So this is at a later time point.
Yeah, so this is the 48. Yeah, okay. Yeah. But still it's very, that it's favorable, and it's also a consistent difference across a lot of different endpoints, which is also, reassuring. And even with SDI remission, you have 25 versus 17%. So this is of course very good. And this is UPA 15 milligram and not 30 milligram. So that's also good.
Right. And do you have any thoughts on how the comparison for upadacitinib versus adalimumab compares to that seen for other JAK inhibitors?
So you said with baricitinib, from the BEAM study, for example, where you had adalimumab as an active comparator? Is that what you asked for?
Yeah, I mean, just thoughts on how maybe the upadacitinib data compares to that seen for other JAK inhibitors. So RA-BEAM, I guess, oral strategy, and then maybe Finch 1 as well.
Yeah. So of course, oral strategy is that was a negative trial. Tofacitinib was not superior to adalimumab. Whereas for RA-BEAM and for this study, you have a differentiation. So that is of course talking in the direction that upadacitinib may be better than adalimumab, but it's again, it is not easy to interpret the clinical importance of this difference. But from exactly this study, it looks pretty good. 36 versus 23, SDI 25 versus 17. But but it's difficult to. It will also very much be. This will have also impact on the how you consider how different countries will also rank these drugs, and that will again also depend on the price. So let's say that if upadacitinib is 3 times as expensive as a biosimilar adalimumab, is that price difference versus the difference that you see here in these data, for example?
Right. Okay. Very helpful, thank you. So on the next slide, there is data from Select Next, now at week 60, so slightly later again. But any thoughts on this data?
Yeah, so that was the patients who also have failed the conventional synthetic DMARD. And here, just a second. Mm-hmm. So this is placebo versus, it's
so patients were switched after the primary endpoint to receive either upadacitinib 15 or 30 milligrams, so they weren't on placebo through to week 60. But the analysis still provided these as split out from patients who had been receiving upadacitinib 15 or 30 milligrams since initiation of the study.
Yeah. So here it seems that, there was no differentiation between 15 and 30 milligrams. Actually, so which is different from some of the data that we have discussed previously. So that's so this again shows that results are not necessarily consistent across different trials. So it's not so easy to really interpret whether there is a clinically significant important difference between 15 and 30 milligrams, for example.
Sure. And then I guess we can move on to the next slide, which is data from Select monotherapy. So, as the study suggests, monotherapy in these patients. So that is the patients who. So this is also 48 weeks. So that's the patients who were randomized. I must just say. So the patients originally were randomized to methotrexate versus UPA versus UPA in 2 different doses. Correct? Yeah. And then at week 14, they were randomized, or the patients who were on methotrexate were randomized to the 15 or 30 milligram dose of upadacitinib.
Yeah, 15 or 30 milligram. Yeah. So it seems, again, there is no major difference for the stringent endpoints across these different arms that you show in this table. Mm-hmm. So, but again, here, 30 milligram dose seems to be numerically slightly superior to the 15 milligram dose, which was not the case in the study that we just discussed before.
Sure. Okay, and then we can move on to the final slide for upadacitinib, which is on slide 13, the data from Select Beyond, looking at week 60 data in BDMARD IR patients.
Yeah, so that's that patients who had failed biologic DMARDs. Mm-hmm. I think this was a study that originally was published in Lancet together with. So as a phase 3 trial. Yeah.
This Select Next, I think, were the first one.
Yeah. So I was actually one of 2 authors writing the editorial for that.
Right.
So when we then reviewed the paper and looked at the paper, we were impressed about the results, and here also you see that, the stringent remission criteria is achieved by a large number of patients, and here the 30 and 15 milligram dose does not appear to be very different from each other.
Sure. Okay. And then kind of a question looking at the phase 3 program for upadacitinib. How do you think the level of data, so not necessarily the quality, but sort of what it's being compared in the patient populations, the comparators, how do you think this compares to the phase 3 programs which are being conducted for other JAK inhibitors?
So I think that I'm really impressed about, the comprehensiveness of the phase 3 program that AbbVie had set up for UPA. So, they have the different relevant patient populations. They have the monotherapy, they have the combination with methotrexate, and they have the active control with adalimumab. So overall, it's a very impressive program for, documenting the efficacy of upadacitinib. But again, still we have these questions about safety and efficacy of 30 versus 15 milligram.
Right. And I'm not sure if you're aware that the upadacitinib was recently approved by the US FDA, and this was only the 15 milligram dose. So I don't know if you have any initial thoughts on that.
No, so I think that makes sense.
Right.
So, but this is again, it is really not very easy to interpret, because as we looked at before, for filgotinib, you don't have any clear relationship between dose or the dose-dependent occurrence of adverse events with 100 versus 200 milligram, whereas 200 milligram seems to be superior regarding efficacy. Here for UPA, you have better efficacy with 30 than 15, so that you have more adverse events. So that's why FDA, I guess, approved 15 and not 30.
Right. And I'm going to ask you about where you would position it, again, in the treatment algorithm. I don't know if this is just going to be the same as before, so it's depending on the pricing. But, yeah, where could you see, if it were approved, upadacitinib fitting into your treatment algorithm?
Yeah, so again, it's also consistent, I think, with the EULAR recommendations, saying that in patients who are intolerant to methotrexate, the JAK inhibitors and the IL-6 inhibitors may have some advantage compared to the TNF inhibitors, for example. So that's where I think it fits into the treatment algorithm. So in patients who start with methotrexate and then fail methotrexate according to a treat-to-target strategy, and then you are going to add something to methotrexate, I think there will not be any major concerns regarding whether you should use a JAK inhibitor or a TNF inhibitor, except that it will be very much dependent on the price. And and what will happen now, probably I'm not sure when the patent for tofacitinib expires, but then you could expect that you will have a generic tofacitinib, which potentially could be very, very cheap. So that's so we will see in 2-3 years what happens with that, for example.
I believe it's 2023, maybe. It's around then. So yeah, not too far off now.
Yeah. No, it's not too far away, but this is always difficult, and Pfizer, they have probably a lot of clever lawyers who are going to try to protect their patents.
Right,
so,
for sure.
Yeah.
Okay, so now if we move on to the next slide, this is looking at some data for asilimab, also known as GSK 3196165, so their GM-CSF antibody. So this was PRO data we saw for the first time at EULAR this year from the Baroque study, which failed to meet the primary endpoint of DAS28 remission at week 24. So what is your opinion on the PRO data available on this slide?
So, when you, first of all, if you fail the primary endpoint in patients population with RA in the phase 2 trial, that's not very promising.
Right.
And because, also in rheumatoid arthritis, the market is so, busy already. You have so many different mode of actions, and now 4 different JAK inhibitors already coming to the market. So if you have a drug, which even if you then have some differentiation for pain, like you see here, it's not very promising if you fail an inflammation-based primary endpoint. So, in my opinion, that will be much more important than whether you have a differentiation for patient-reported outcome measures.
Right. And, I mean, taking that into account, obviously, I know you mentioned pain. During the presentation at EULAR 2019, GSK stated that GM-CSF may be involved in the pain response as evidenced by inhibition of pain in preclinical models following inhibition or absence of GM-CSF. Do you think the data shown here supports this assumption?
Difficult to know, because, the patient global will very much be driven by pain, and I must say I don't know. So they also, as is said here, you also saw improvement in fatigue, and you saw improvement in the 8 domains of SF36. So it's difficult to say.
Sure. Okay. So then, if we look onto the next slide, I'm not sure if you can make out the details of this, but this is the phase 3 program for otilimab in RA called Contrast. I can run through some of the details if you want, or I'm not sure if you're familiar.
Yeah, that's fine. Yeah.
So, sorry, did you want me to run through some of the details, or?
Yeah, that would be nice, because it was not so easy to read.
Yeah, no, exactly. So I guess one thing is the dosing here is the 150 milligram and 90 milligram weekly dose, which is compared to, in the phase 2 study, there was doses up to 180 milligrams, but these were administered every other week. So that's one thing that they've identified from a phase 2A pharmacokinetic study showing suboptimal trough concentrations, which they've changed. And then one other thing is that they have chosen to go with ACR20 at week 12 as the primary endpoint, following which patients randomized to receive placebo will be re-randomized to otilimab or active comparator. The final thing which I'm going to call out, this might be able to make out from the images, but active comparators are used in all 3 of the studies. So 2 of them are utilizing tofacitinib, and then 1 of them is utilizing sarilumab as an active comparator. So, starting off, what is your opinion of them using ACL20 at week 12 versus placebo as the primary endpoint for this program?
I think it's becoming more and more unusual to use ACR20 as the primary endpoint for superiority trials, because again, as I said before, ACR20 is, a regulatory endpoint to differentiate between active treatment and placebo. And for a clinician, ACR50 or 70 is more important. So ACR20 is okay for biosimilar trials to show equivalence or non-inferiority. But for a superiority trial, I would have expected to have a more stringent endpoint as a primary endpoint.
I mean, they're obviously evaluating all of them, but as secondary endpoints, as you say. So what is your opinion of the dosage and the frequency of dosage being changed from the phase 2B study to this phase 3 program?
Oh, that must be a risky thing.
Right.
So, because usually in a phase 2 trial, you also, test different doses, and then you identify a dose which you also then use in the phase 3 trial program. So if you have a phase 2 trial failing the primary endpoint, and then you reduce the dose in the phase 3 program, that must be a...
So they increase the frequency of dosing from every other week to weekly, though. So I don't know if that's...
Okay, sorry. So the total dosing should be the same.
So they went from up to 180 milligrams every other week in the phase 2B program, and in this, the higher dose is 150 milligrams weekly.
Okay, so then I misunderstood. Sorry.
Yeah, so, to make that very clear.
Yeah, okay. So, no, but it's no. So then I understand. That's more logical.
Right.
So, No, so that makes sense, and I think that they have also an active comparator with a JAK inhibitor and also an IL-6 inhibitor. That's interesting, and it's also a new approach actually, because usually adalimumab has been the standard drug for active control arm.
Right. Well, I was going to ask you on your thoughts for the head-to-head inclusion of tofacitinib and sarilumab. So if you have any other comments, or we can move on to the next question.
No, I don't think so.
Okay. Yeah. What do you think about them not including adalimumab as a comparator? Do you think that is a mistake, or is that fine for your opinion?
So it's I don't know why they did it, but they probably had some reasons for it. So but then this has also been negotiated with FDA and EMA, so they have probably also then accepted that. So I must say I don't know.
Okay.
Why. Yeah.
Sure, that's fine. So now, kind of again looking at the trial design, Contrast 3, so this is the one in BDMARD or JAK-IR patients against sarilumab, includes JAK-IR patients in the study population. How important do you think it will be for therapies to have data from this patient population as more JAK inhibitors are used in RA?
So you said this is the Contrast 3 trial where you have previously inadequate response to biologic DMARDs or JAK.
Yeah.
And or JAK inhibitors. So I think so. That's why they that's probably why they selected sarilumab for this trial, because if they had a TNF inhibitor, if you have then previously inadequate response to a biologic DMARD, then you couldn't use. It wouldn't make sense to have adalimumab as the active comparator. So and also if you have failed a JAK inhibitor, it doesn't make sense to have a JAK inhibitor as the active comparator. So that's why they probably selected sarilumab. Mm-hmm. No, but I think in general, you need to do trials in methotrexate naive patients, methotrexate failures, and biologic DMARD failures. And in this case, either biologic DMARD failures or JAK inhibitor failure, if I understand it correctly.
And then, so GSK is not evaluating otilimab in methotrexate naive patients or as a monotherapy. What's your opinion of this?
Difficult to know, but again, they probably have their reasons for that based on some post hoc analysis of the phase 2 trials or something like that, or if it's something related to the mechanism of action or mode of action, I don't know. But in general, as I mentioned, the biggest gap in the need from the healthcare system is patients who are failing or are intolerant to methotrexate, and that's a significant patient population.
Okay. And then does the inclusion of novel comparators, so tofa and sarilumab, in these head-to-head studies suggest that we might see more head-to-head studies with novel comparators in the future?
I think so. Mm-hmm. So this is, as far as I know, the first time that you have something different from adalimumab. But as we discussed recently, that since you have in the inclusion criteria or enrollment criteria, you have failure of biologic DMARD and or JAK inhibitors. Of course, then you need to have an active comparator with a different mode of action.
Sure.
So I assume that previous failure of IL-6 inhibition will probably also be an exclusion criterion in this trial. I guess so.
I would imagine so. This is the one which is not available on clinicaltrials.gov yet, so don't have the full details for this. The other two are available. And then, it's, again, a slightly speculative question, but what head-to-head studies would be most beneficial for you to assess the current pipeline and or marketed therapies in RA?
So I think that, if you are going to, with the limitations that exist, if you are going to try to compare trials with each other, So in patients who have methotrexate failures or methotrexate failures, then I think adalimumab still should be the standard active comparator because, when you fail a methotrexate, then, the treatment option would be a JAK inhibitor or a biologic DMARD drug, and adalimumab is the most widely used TNF inhibitor globally. So, and you now have, like in Europe, one was withdrawn, so there's still 4 biosimilar adalimumabs available. So in many ways, there are strong arguments for using adalimumab as an active comparator in a population who has failed methotrexate.
Sure. Okay. So now moving on to our final section for today. This is looking at some phase 2 candidates. So starting off, we've got BioCAD's BCD089, which is an anti-IL-6 receptor antibody, and this is data from the phase 2 Aurora study. So do you have any thoughts on this data?
Mm-hmm. I looked at it before we started. This is a very small phase 2 trial with only 35 patients in each treatment arm. And, you have ACR 70 responses of 28, 20, and 3%. So it's a clear differentiation between the active drug and placebo. Also for ACR 50, 51 versus 5%, ACR 20, 77 versus 17. So at least for the highest dose, it seems that so that is the which means the weekly dosing. It seems to be pretty convincing data actually, and also for low disease activity, DAS28 CRP 57 versus 3%. Mm-hmm. But I think that from this small study, it seems that weekly dosing will be required.
Right, and that was one thing that they said in the poster as well, is that for the phase 3 program of this drug, it will be evaluating the weekly dose of BCD089. So do you have any thoughts on how that compares to other IL-6 inhibitors?
This was in combination with methotrexate, and again, here it will also be very important in the phase 3 program to evaluate monotherapy. Mm-hmm. But again, it's a competitive market. You have tocilizumab, you have sarilumab, you have this other drug that was withdrawn from the market. So it was
Erizumab.
Yeah, exactly. Yeah. So no, but if they can find, so again, in this competitive, because here for IL-6, you have this, also competition between the JAK inhibitors and the IL-6 inhibitors, as I mentioned,
right.
In the EULAR recommendations. It seems that, these drugs are seen, more or less on the same level because they are working very well as monotherapy. But, it's a small study, but for weekly doses, it's a very clear differentiation, clearer differentiation than usual to placebo in combination with methotrexate.
Right. Okay. And if we can move on to the next slide, this is data for fenebrutinib, which is Roche and Genentech's BTK inhibitor. So do you have any thoughts on this data?
Yeah. So this is less, it seems to be less promising in some way, because if you look at the week 12 results, so then you have, if you again look at ACR 70, you have for the new fenebrutinib, you have, 13% ACR 70, and 18% for adalimumab, so numerically lower than adalimumab, and the differentiation, here between the active drug and placebo is much less than we saw with the IL-6 inhibitor from BioCAD, the BCD089.
Yeah, and obviously the powering for this study was just looking at p less than 0.2. Do you have any comments on that as well?
Once more, you said.
Sorry, the p-value for this study to show significance was just 0.2. Do you have any comments on this?
Yeah, that's also very low, and but I think it's sometimes instead of looking at the p-values, it's more important to look at the magnitude of the response.
Sure.
And again here, you have a lot of patients. This is a larger phase 2 trial than we saw with our 6 inhibitors. So here you have 110 patients in the highest dose, compared to 110 in placebo also. And the differentiation for both ACR20 and the more stringent endpoint is not very strong. And adalimumab is numerically more effective than the new drug. So,
sure.
So if I was the CEO of Roche Genentech, I would have stopped this development.
Okay.
Do you know if they go further to phase 3?
I'm not sure. We don't have any information about that at the moment.
Oh, okay, yeah.
Are you familiar with combinations of BTK inhibitors? For example, AbbVie is evaluating a BTK inhibitor in combination with upadacitinib, so BTK plus JAK.
No.
Okay. Do you have any comments on whether you think this would be a viable strategy in RA, or no comment?
I must say that I don't know, because that depends also very much on the safety profile, I think, because, this combination of different drugs with different mode of actions, there have always been some issues with safety when you do that. But if you have some, clear that you have data supporting that there should not be any added safety issues by combination, and that you have different mechanisms of action, so that you could have not only an add-on but a synergistic effect on inflammation. So then might do you know if a phase 2 trial is carried out with the combination?
So there's a phase 2 currently recruiting ongoing, but no data available yet for that.
Okay, yeah. So we will see.
Sure.
But of course, combinations with the different mode of actions, that is, of course, always interesting. But again, as I mentioned, there could be safety issues.
And then the final slide today is on Eisai's E6011, which is an anti-fractalkine MAB. Do you have any comments on the data on this slide?
Yeah, so here also you see that the ACR70 responses are not very different from placebo, and neither there is some differentiation at week 24, but at week 12 it seems to be very similar, and also for ACR50 you have 18 versus 15%, and for 200 milligram dose. So I don't think this looks very, very promising, actually.
Sure. And we have very little time left, so I'm just going to ask a couple of general questions, if that's okay. So the first one is, are there any other phase 2 or early candidates in RA that you're familiar with that you think could be promising for the potential future treatment of RA?
No, I must say, not to my head today, but as I mentioned before, that the market for RA is, of course, rather tough, because you have now many different mode of actions, and except for this differentiation for baricitinib and UPA and filgotinib against adalimumab, there is overall, you have more or less the same magnitude of response for the different drugs, both in the JAK inhibitors and the biologic DMARDs. So it's a bit surprising that when you have all these drugs, that they more or less have the same magnitude of response. So it's a tough market.
Sure. Well, that's all we have for today. So I want to thank you for your time and your responses. It's been very helpful, and we'll send through the honorarium shortly.
Okay.
Thank you, and have a good rest of your day.
Thank you. Same to you. Bye-bye. Bye-bye.